Hello everyone. We would like to present on genetic variants associated with vitamin K deficiency. Vitamin K is one of the fat-soluble vitamins found in foods and marketed as dietary supplements. The human body requires vitamin K for post-synthesis modification of certain proteins that are required for blood coagulation for controlling binding of calcium in bones and other tissues. It has a family of highly lipophilic molecules and there are three different forms of vitamin K. It includes two natural vitamins, vitamin K1 and K2. Vitamin K3 is a synthetic form of vitamin K, was used to treat vitamin K deficiency, but because it interferes with the function of glutathione, it is no longer used this way in human nutrition. Vitamin K obtained from the diet is considered to reach the target tissues via lipid absorption and the transport system. Once transferred to the cells of the target tissue, vitamin K is metabolized by redox cycling in the intracellular endoplasmic reticulum body in a process known as vitamin K cycle. Warfarin, an oral anticoagulant drug, inhibits VCAR, stops the vitamin K cycle, and prevents gamma glutamyl carboxylated conversion of blood coagulation factors, thus inhibiting coagulation. A total of 46 vitamin K related genes are predicted so far, and it plays a major role in bone development and tissue calcifications. Genes are associated with a number of comorbidities such as cardiovascular disease, chronic kidney diseases, lupus, and liver diseases. The key genes of vitamin K pathway are VCAR-C1, CYP2C9, CYP4F2, GGCX. CYP2C9 and VCAR-C1 are the primary genes known to influence warfarin dosing. These candidate genes are responsible for bleeding, thromboembolic events, and warfarin dosage.
For this analysis, we have analyzed a total of 3 RNA-seq datasets: myocardial, prostate, and renal, comprising of 19 samples. Finding out SNPs from RNA-seq data is the challenge that we tried to take during this work. The first step of this analysis is to check the quality. This was done using FastQC. Next step was to align the raw data to the human genome reference. This was done using HiSet2Mapper. Variants were then called using SAMTools MPileup option. The called variants were then checked against the already published and validated list through ClinVar database. We found about 114 variants that included false hits. After filtering out the false hits, we were able to identify a total of 3 variants corresponding to ATP81, CFTR, and SIN1 genes. These variants were then checked in Genome8 database for allele frequency validation. The ATP81 variant is clinically associated with coagulopathy. The variant of interest in CFTR gene has not yet been reported by clinical diagnostic laboratories, although HGMT cites the variant as disease causing. The SIN1 protein plays an important role in the maintenance of the brain region that coordinates movement. This particular variant is reported as a missense variant. All the presented work has paved a way for us to discover valid SNPs from RNA-seq data. The same workflow can be applied to any given RNA-seq dataset and a set of comorbidities. We would like to thank Dr. Prashanth Suravajhala, Dr. Jalaja, Dr. Anil Kumar, Dr. Praveen Mathur, Dr. Anita Simlot, Dr. Sudhir Mehta, Dr. Chagan Bihari, Dr. Sumita Mehta, Mr. Ashwani Kumar Mishra, Dr. Krishnan Mohan, Dr. Bhanu Prakash Reddy, Dr. Kavi Kishore for their constant guidance and support. Thank you all for your patience.